Welcome. In this video, we're going to discuss the concept of long-term potentiation. So long-term potentiation is thought to be a mechanism behind learning and memory. It involves changes between the strengthening of connections between neurons, specifically strengthening synapses following repeated stimulation. So you have one or more axons that bombard a dendrite with stimulation, and that's what we're seeing here, right? You have a neuron, this is the dendrite receiving information, and you have multiple axons bombarding it with stimulation. And remember, those cells that fire together wire together. As a result, you're going to see that receiving cell becomes potentiated. So here's normal stimulation. You have the presynaptic cell, you have the postsynaptic cell, and you have this depolarization, remember this EPSP in the postsynaptic cell. Well, after long-term potentiation, when that axon's been bombarded by one or more axons with lots of stimulation, it becomes potentiated, so that the depolarization is greater. So again, the synapse becomes potentiated, or more responsive. Or as it's saying here, the synaptic strength increases. And this is just another diagram kind of showing the same thing. You have your postsynaptic neuron with the dendrite receiving information. You have strong stimulation from axon 1, weak stimulation from axon 2, no stimulation from axon 3. So these two firing together strengthens the synapse. Again, very similar to what we were talking about with associative learning. So now your response, this is your response, your baseline response, and now it's potentiated or enhanced. We'll talk in more detail on a subsequent video, but the biochemical mechanisms of LTP, what's happening at the biochemical level, at the synapse level, is that there's changes in glutamate synapses, or at glutamate synapses, primarily in the postsynaptic neuron, but we do see some changes in the presynaptic neurons as well. So the main glutamate receptors involved are going to be your AMPA receptors and your NMDA receptors. So remember, glutamate receptors are named after different neurotoxins that activate them. So AMPA and NMDA are different types of toxins that activate those receptors specifically, and there's also a third one called kainate. There may be more that I'm not aware of. So again, this is just showing you at resting potential, you have your presynaptic cell releasing neurotransmitter, which is glutamate. It's going to bind that AMPA receptor, go in and cause a depolarization. As we'll see when we talk more in detail about the biochemical mechanisms, just one firing of an active potential isn't enough to cause LTP, because it involves both the AMPA and the NMDA receptor. And we see here the NMDA receptor has what's called a magnesium block. So you need repeated stimulation. And when you have that repeated stimulation that you see here, you have a bigger depolarization in the postsynaptic cell that repels that magnesium, that allows calcium to come inside, and then that's going to cause some downstream effects that we'll talk about in another video.